Hi, good morning, all.
Good
Good
morning,
morning,
sir.
sir. Morning, sir.
Mm. So we are meeting after I think more than a week. Actually, it's been quite very how to say, very confusing and very puzzled kind of a scenario nowadays. We yet don't know the CUSAT admissions have not yet finalized. So I don't know how many new candidates will be joining in more. The CUSAT admissions are still going on, so it's a bit of a haphazard and a confusing kind of scenario. But we'll try to go on with the course of the lectures, and then we'll see how the new students will cope up with it. Are there any doubts or issues with the video lectures I shared last week? Before I move
Yes,
on,
sir.
sorry.
Yes, sir.
Yeah, tell me.
So,
But you never
I am unable.
asked you guys to post your doubts on the group, actually, so that others can also get to know, and then they can at least try to look up some stuff. Tell me, okay, if there is one question, tell me, but if there are more, then we'll have a different interaction session where everybody can would have gone through the videos, and then we can address the questions of all, actually. Anyways, what it is, you just tell me for now.
So,
So,
as you said in the
Hmm?
lecture, you said that uh, I don't able to get the difference between the code transformation and the binary vector strategies, because you said in code transformation that we use the two vector
Hmm?
simultaneously to uh, transfer the tDNA. Hello, sir.
In code transformation, but when I talked about the binary vector, we did not talk about code transformation, isn't it? With the code transformation was part of the selectable marker, the removal of selection marker genes.
No, sir. You said about it in the lecture.
Okay. Which slide it is, actually? Hang on, then let me
Sir,
open.
can you once again discuss the whole these two strategies?
Then we do one thing. You let let others also go through the video lectures. I don't think so others have gone through. Just give me a second. Yeah, so what I was saying, then in that case, let others also go through the videos, and we will keep the coming Friday class. We'll keep for the doubt and the for the discussion and the doubts and all. Is that all right with all? So you have
Okay,
roughly
sir.
2 days time to go through the video lectures, even the previous videos that I have shared. If you have any queries, any doubts related to that, we will keep the Friday aside for that. Actually, we'll keep the Friday aside for that. All right. and I will still say, see, I'm not able to recollect what are you referring to. So that's why I would still suggest that if there are any specific queries, please try to post it in the group so that I can go through them before I get into the class and know like what you guys are talking about or what you guys are referring to.
Okay, sir. Sir, do you prefer refer any book for the plant
Yes,
biotechnology?
I would refer. I refer to is a book called Biochemistry and Molecular Biology of Plants by Buchanan. I think I'll have a soft copy. I'll share the same with you guys. I'll put on the classroom.
Okay, sir.
Yeah. So I refer to that book, and then of course the internet content is there, or there are like some review articles if you do not find the updated stuffs in the books. But for now, I think you can refer to that book because it's not a very old edition. It's a relatively newer edition. I think 2013 or 2014 or something. So it's not a very old version. So you can refer to the book. It's a Biochemistry and Molecular Biology of Plants by Buchanan, I guess. Yeah. So I have the soft copy. I'll share the soft copy in the classroom. And I will still reinstate or reinforce that if you have any specific queries, please try to post it in the group so that I know what you guys are referring to, so that I can be ready well in advance. Because now I kind of vaguely remember what you are saying, but I'm not very sure which particular slide you are referring to.
Okay.
All right. Yeah. So with that, we'll move on to today's lecture.
Excuse me, sir.
Huh?
Sir, actually, I tried to watch the video, but in some instances, your voice is breaking. So I'll suggest one thing. Can you please share the slides or PPTs?
Slides,
That
we do not. Is this the problem being faced by all? Because I don't think so. The voice is breaking
Yes,
in the
sir.
video lectures.
Yes,
Huh?
sir. Yes, sir.
What?
Your voice is not clear in the lecture at some times.
That is something I don't think so. Because I remember recording the videos and I have gone through the video. Okay, let me go through the video again. Let me go through the video again. I have come up with a strategy of not sharing the PPTs actually, because what happens then, the students, they never look into the video lectures and they just wait for the exam to go through the PPT. They never. So that's why we have stopped giving PPTs. Actually, the PPT is there in the video lecture if I'll go through the videos again, but I don't think so, except for because if the voice is breaking, I have tried to repeat it. If not, we can
Sir, even today the voice.
even today the
So
voice
even
is
today your voice is breaking.
this is something I
Yes, sir.
cannot help. Actually, this is something I cannot help. I have attended many meetings on that. I have taken most of the video lectures using this internet connection. I am not using my mobile phone. It is a Tata Sky broadband connection with 50 Mbps speed. So if that is not able to support the video lecture, then that's something I cannot do. This is something I cannot help you guys with. So if there are points, if there are topics where there is no clarity or there are doubts, feel free to put it in the group. I'll discuss it. You have I will share the book. You guys can go through the book, the lectures, the chapters from the book, and of course you are free to access the internet to get the content from wherever you can find it actually. But
okay, sir. Actually,
we did not have very good experience with in the past where we shared the slides. The students never go through the video lectures until the end of the examination. Until we announce that there will be a first internal examination, the students don't even bother to look at the video lectures. And then I mean the entire the entire process of teaching them, or the entire process of sharing video lectures becomes null and void actually. That's why I have stopped giving PPTs actually.
Okay, sir, no problem. You please share the book, that will be great.
Yeah, I'll share the book. If at points where my voice is breaking, I'll go through the video lectures. And if you also feel that at points where the voice is breaking, you raise that particular topic, or you mention that particular point or particular topic in the group, and I'll try to discuss that again.
Okay, sir.
Yeah, and if it is breaking today also, just let me know. I'll try to repeat the point, but I can't do. I mean, this is something that is beyond my control actually.
Today it is fine, sir. Actually, yeah, it is better than
You guys
when
decide
you are in
yourself whether it is a problem with my internet connection or your internet connection. Somebody is saying it is fine and somebody is saying it is breaking. So I don't know, it's a problem with my internet connection or your internet connection.
But like not much, but sometimes it is breaking.
If you feel that something important has been missed, just let me know. I'll try to repeat the part actually.
Okay, sir.
Yeah. So if getting back to today's bit, if you guys have gone through the course structure in like recent times, can you see the slides? So there is a topic called I have tried to cover the agrobacterium mediated plant transformation and how you generate and characterize transgenics. Then there this removal of selectable marker genes was also there, and then there is topic called screenable and selectable marker genes or selectable markers. So I mean, this is I consider it quite important with reference to, or with respect to today's modern day plant biotechnology, because whenever we generate transgenics in lab, we do use selectable marker genes. Whenever we generate transgenic in lab, we do use selectable marker genes. And of course, also we also use screenable marker genes actually for modern day plant biotechnology experiments that we do. So I thought it's better to cover this topic by devoting a couple of slides to it actually. So selectable markers are basically, it's a marker gene that encodes a product that allows us to select the transformants from the non-transformants actually. So what it will does, it provides, it helps us to select the cells that are transformed with the transgene that we are introducing, over the cells that have not received the transgene. So it will provide, I mean, are you guys getting it? Have you heard of antibiotic resistance?
Yes, sir.
Yeah. So
Yes, sir,
selectable marker genes are very much the antibiotic resistance genes actually. So you have heard in bacteria, whenever we try to select the bacteria, we have an antibiotic resistance gene like ampicillin or kanamycin. So what it does actually, it helps to select the transformed cells from the non-transformed cells. So we grow the bacteria after doing the transformation. We grow the bacteria on a agar plate containing antibiotic. So only those cells which have the antibiotic resistance will be able to survive, and the ones that do not have the antibiotic resistance will not survive. So this is a very, very basic concept that you guys see while doing bacterial transformation. But we utilize the same concept when we do plant transformations. This is just an example over here. You see, when you place the seeds, allow them to germinate. These are a pool of seeds containing transformants and like the transgenics and non-transgenic plants. So the transgenic plants will have resistance to certain antibiotics or certain herbicides, and the non-transformants will not have any such resistance to the herbicides. So when we grow them on a herbicide containing plate, the transgenics will survive. All these green ones will survive. I mean, the ones that you see as green are the ones that have the transgenic, that have the antibiotic resistance or the herbicide resistance. They will survive and the others will die off actually. So these are the marker genes that provide resistance to antibiotics or herbicides, and I have listed out some of the very commonly used herbicide resistance or antibiotic resistance that is used in plant transformation systems. So what happens actually, if you would have gone through the video, you would have seen the binary vector or the vector that we use for plant transformations. So it has the LB-RB, the left border, right border region, and you have the gene of interest in there with the promoter region and the terminator region. And we have an additional transcription cassette. We call it a transcription cassette. So it will have a promoter region, a herbicide or antibiotic resistance gene, and a terminator region. So there are basically two transcription units that are present between the LB and RB, the left border and the right border regions. And then there is another antibiotic resistance gene that is present outside the LB and RB. So this is how a vector is actually designed. So and of course you will have origin of replication and things like that. So the antibiotic resistance gene that is present outside the left border and right border is required for cloning and moving it into agrobacterium. So that provides resistance against the bacteria in the bacterial system. So it will have a bacterial promoter. So that T7 promoter or a bacterial promoter that will be active only in the bacterial system. So that is the antibiotic resistance that is required for cloning purposes or for introducing them into agrobacterium. But once the agrobacterium has been utilized to infect the plants, the tDNA gets integrated into the plant genome. Now your this antibiotic resistance outside the tDNA or outside the LB-RB is no longer useful. It is gone actually. So then we need to have an antibiotic resistance that will help us to select the transformed plants or the transgenic plants. So that's where this antibiotic, which is present between the LB and RB, gets into picture. And these are some of the very, very common examples that we use. One is called NPT or neomycin phosphotransferase. One is hygromycin phosphotransferase. Neomycin phosphotransferase is the gene that provides resistance to. Any idea? If you don't know, you can just say no actually. So
No, sir.
yeah, so yeah, it's okay to say no. I do not expect you guys to know everything. Otherwise, what's the point of me putting things here actually on the slide? I just try to check like if anybody have read anywhere or if anybody have heard of it. So neomycin phosphotransferase is the gene that provides resistance to kanamycin. Have you heard of the antibiotic named kanamycin?
Yes, sir.
Yes, sir.
Yeah. So neomycin phosphotransferase NPT2 is the gene that provides resistance against kanamycin. Hygromycin phosphotransferase is the gene that provides resistance against hygromycin. Hygromycin is another antibiotic that we use for plant transformation purposes. Then, if you have heard of the gene called Barster gene, the Bar gene, if you have heard of the word called Bar gene, resistance to Bar gene. So this is phosphorinothycine acetyltransferase. This provides resistance against Bar gene. And then there is a very famous one called Roundup. Have you heard of a herbicide called Roundup? We use it in the. It's a very commonly used herbicide in gardens actually.
Yes, sir.
Yes, sir.
इसके बारे में सुना है? राउंड अप के बारे में सुना है? लोग स्प्रे करते हैं जहां पे अनवांटेड ग्रोथ
Yes,
वगैरह
sir.
आता है, फार्मर्स भी यूज करते हैं. राउंड अप इज अ वेरी वेरी
Yes.
कॉमन herbicide that is used actually. So that is another yeah, so Roundup is another herbicide that is used quite commonly for generating transgenics. And Roundup is a herbicide. It is an analog. It blocks the shikimate pathway. Now, what is the shikimate pathway known for?
Right, in metabolism.
What
Yes.
metabolism? What metabolism?
Yes,
Nucleotide
plant
metabolism.
metabolic pathway.
नहीं, plant metabolism से इसका वो नहीं है. There is a
Yes, sir, no, sir, sir.
है?
Nucleotide metabolism.
Microorganisms and plants both. So for the biosynthesis of amino acids and
What amino
folates
acids?
and
What amino
sorry,
acids?
aromatic
Yes, exactly.
So,
aromatic amino
This is
yes, sir.
this is what I was looking forward to. Shikimate pathway is a very recognized, well-known pathway that is involved in the biosynthesis of aromatic amino acids. And Roundup blocks this 5-enol pyruvyl shikimate phosphate synthase. It blocks this particular enzyme, so it blocks the shikimate pathway, and as a result the entire plant dies in a couple of days. Actually, in 2 to 3 days you will see the plant start wilting and it will die off. So these are some of the examples of the different selectable markers that are very routinely used. Among these, neomycin, hygromycin, and bar are very commonly used, like more often than not the vectors that we use in the lab, they have one of these three selections, antibiotic or herbicide selections. Actually, I've just tried to list out a series of selectable marker genes and their source and the principle of selection. What resistance do they offer? So you can see here the NPT2 provides resistance to neomycin or kanamycin, then HPT against hygromycin, phosphorinothrycin acetyltransferase against bar. This is glufosinate, or it is called BASTA actually. Yeah, we use it is by the chemical name BASTA. We use BASTA resistance. And bar stands for BASTA resistance. So the BASTA is the herbicide. Then EPS, PS, I have already told, Roundup, and then there are a couple others actually. Now coming to the screenable markers or the reportable or the reporter markers or the reporter gene markers actually. So there is one way of doing it is to select them on the selectable markers, but of course they provide resistance against the antibody. So for under certain circumstances, people are not very comfortable using selectable markers actually. So then they go for screenable markers or the reporter genes. In many cases, we also use reporter genes. So I mean, these examples, I am sure you must have heard about it. You must have heard about GUS and GFP and luciferase. So these are some of the genes that are used as the reporter genes or the screenable marker genes. Now,
Yes, sir.
screenable marker systems, they employ a gene whose protein product is easily detectable. Now here you are not selecting it. Now one of the issues sometimes that people feel with selectable markers is that they provide herbicide resistance. So sometimes when you want to apply herbicide, or when you want to use it for certain purpose, you cannot use, because the non-transformants will die without you want them to be losing it. So these are some of the unique scenarios or specific scenarios where you don't want to have a selectable marker gene in your transgene. So that's why, and then there is also a possibility, like people say, that sometimes it can lead to some unique recombinations within the plant and can develop such a variety that cannot be killed by any of the by any of the herbicides or antibiotics. So this is not acceptable, because we are kind of manipulating the nature. So that's why sometimes people are not very comfortable with selectable marker genes. So the other option that we have is having a reporter genes. Now reporter genes, besides being used for screening purposes, it's not a selectable marker gene, it's a screening marker gene. So, but besides being used for screening purposes, it is also used as a very powerful tool for many kinds of for reporter gene studies that we do. I'll come to that actually, I'll come to that. But just to start with screenable markers, they employ a gene whose protein product is easily detectable in the cell, either because it produces a visible pigment or because it fluoresces under appropriate conditions. Now, visible markers, they rarely affect the steady trait of interest. They are not adding any additional trait like the selectable marker genes, you see. So, but they provide a powerful tool for identifying transformed cells before the gene of interest can be identified in the culture. Now, what I was trying to say, so these are some of the examples like luciferase, GFP, and GUS. What I was trying to say, that if you have a pool of seeds where some are transformed and some are non-transformed, and you put them under a fluorescence microscope, you provide them the required wavelength. So, and say the transformed or the transgenic construct has a GFP protein in it, actually is fused to your gene of interest. So if your gene is getting translated into protein, the GFP will also be there. So what will happen when you fluoresce it with a particular wavelength, this GFP starts fluorescing. It will start giving signaling. So this GFP will start this GFP will start fluorescing, and if you look under a microscope, you can separate out the GFP signal seeds with the non-GFP signal giving seeds, actually. So that's how you are able to screen without any special antibiotic or without any special chemical. You are able to screen the transformants from the non-transformants. Now that's how you can use a screenable marker gene, actually. Now this is for screening the transformants from the non-transformants without any additional antibiotic or anything. But what is more important, as I mentioned, that it can also be used as a tool to identify in what particular tissue or in what particular cell your gene is getting expressed. You see, for these are some of the examples that I have listed here. Now, say I'm working on a protein which is a relatively new protein, nobody has worked on it, and I feel that that particular protein is a transcription factor and it is nuclear localized. So it is a common practice that whenever we try to characterize a new gene or a new protein, we do a subcellular localization study and a tissue specific localization study for that particular gene to get an idea that what kind of function it might be doing, or what is the developmental aspect, or what is the role of that particular protein. Say if I believe my gene my protein is a transcription factor. So if it is a transcription factor, it should be expressed inside the nucleus, because that's where all the transcription factors work, actually. So what I will do, I will fuse it to the GFP protein, and we'll check for the subcellular localization of the protein. So if you see here, these are all the chloroplast, and you have the cell and everything. I mean, this is how the picture is taken. So you have the nucleus over here, and you can see the GFP signal coming from the nucleus. And in order to further confirm it, we stained it with DAPI. I mean, in this picture, you see it has been stained with DAPI. So DAPI intercalates with the DNA. So it will light up the nucleus. So if the GFP signal is coming from the nucleus, the two signals will overlap, and this is how it looks like. So this confirms that the protein that I am working on is actually expressed in the nucleus. It is a nuclear protein. So this is one of the benefits of screenable marker genes, actually. Similarly, this is an example of GUS, actually. Say I have a protein, the same protein, and I mean, this is the subcellular localization. This is a tissue specific localization. And I have a hypothesis that that particular protein have a role to play in seed germination and dormancy. So what I did, I used the gene and I fused it to the GUS. So wherever the gene is getting expressed, the GUS will also get expressed, actually. So GUS will also, so now we have the different tissues from the plant. We have the inflorescence, we have the flower, we have the seed, the non-germinated seed and the germinated seed. And we have the thing, and you take them individually and you do the GUS staining, the experiment. You treat them with the, so GUS is actually, as you can see here, GUS stands for beta glucuronidase, and this is an enzyme that breaks down the substrate X-alpha-gal. X-gluc, sorry, not X-alpha-gal, X-gluc. It hydrolyzes the beta glucuronides or the X. So we have a substrate for X-gal, for GUS, and in whatever tissues this GUS enzyme is getting expressed, you just take the different tissues and you do the GUS assay where you provide the substrate. So if the GUS enzyme is getting expressed, it will start breaking down the substrate. And once the substrate is broken down, it gives a blue color. So we do a decoloration process where we get rid of all the chloroplast and everything, so that this color becomes very prominent and distinct actually. So once after decoloration and all removal of all the pigmentation, this is how it looks like. So this is how it looks like. So this indicates that our protein is actually getting expressed in the anthers or in the germinating seeds. So this is how you use the GUS to identify the specific tissue where our protein or our gene is getting expressed. So this is a very, very important these screenable marker genes have a very, very important role to play in modern day plant biotechnology. We use these GUS, we use them as screening markers, or more importantly for tissue specific localization and for subcellular localization quite routinely in the lab actually. So talking about the selectable markers and the screening markers, the other point that I want to start on, I don't think so I'll be able to finish it now, because I think this is ending. We'll try to take it as far as we can. Oh, you guys, as you have another class at 12:30.
Yes, sir.
Okay, so I'll try to get started with chloroplast transformation. We'll cover some of the slides, and then so you guys need to join back again in the same link actually, because this link is about to finish. Okay, so you guys join back using the same link, and we'll try to go like for the next 15 minutes or so, and then you guys can leave for the next class. So there is another topic in the syllabus called chloroplast transformation. So I hope you guys are quite aware of the chloroplast genome and all, but I have just in order to brief it, I have just listed down some points which are relevant to the chloroplast genome. And in the next slides, in the following slide, you will get to know that why chloroplast transformation is important, like what we have studied so far using the agrobacterium mediated or the direct gene transfer method is nuclear transformation. So the nuclear genome is getting transformed, the nuclear transformation, but an important aspect that is coming up is chloroplast transformation. So I feel that it is good to cover this as well actually, I mean, not letting it go. So talking about the chloroplast genome, they have their own genetic material. We all know that they have been derived from the prokaryotes and all, with about 300 plastids in one cell. And we also all know that chloroplasts are maternally inherited, and it ranges about 120 to roughly about 120 to 150 kb is the genome size, and which encodes about 120 proteins that are responsible mainly for gene expression, photosynthesis, and metabolism. It's a circular double-stranded DNA, and it consists of the double-stranded DNA is comprised of a large single copy, a small single copy, and a duplicated inverted. There is an inverted region, IR region in the chloroplast genome, which is replicated or duplicated, not replicated, which is duplicated n number of times, from 1000 copies to 10,000 copies of the genome can be present in a single cell actually. So this inverted regions, the IR regions, are duplicated n number of times. That's why the genome is so big, but it only encodes for a very small number of protein, because these are all duplicated events. Now, why chloroplast? Why people started focusing on chloroplast transformation when nuclear transformation was going on so well actually? It acts as a direct source of, like, chloroplast is the place where carbon sequestration and starch production takes place. So that's why it acts as a very, very important tool. Chloroplast transformation acts as a very important tool when we want to monitor, or when we want to play or manipulate the carbon sequestration process or starch production process. I mean, talking about carbon sequestration, I don't know if you guys have heard of it or not, but there is a very, very interesting project going on in the world, being led by IRI, International Rice Research Institute in Philippines, Manila, where they are trying to convert a C3 plant into a C4 plant. Because C4 plants are more energy efficient, they do better, they have better rates of photosynthesis with minimal loss due to the photorespiration and all. So that's why. Okay, so yeah. So what I was saying, that talking about carbon sequestration, there is a very interesting project going on where scientists want to convert a C3 plant into a C4 plant because they are more energy efficient. So this is so that's why for some of these things where because chloroplast is the site for starch production, carbon sequestration, and amino acid synthesis. So people thought that it might be a good idea to target directly the chloroplast for transformation rather than doing nuclear transformations. And then because chloroplast has been derived from the prokaryotes, it does not have a single gene concept, rather than it relies on the polycistronic mRNAs. It has polycistronic mRNAs where several genes are being governed by one operon. So when you do a transformation in chloroplast, the protein levels, the protein accumulation or the protein synthesis is much, much higher as compared to the nuclear transformation. And because it has the feasibility of, and it also has the feasibility of expressing multiple proteins from polycistronic mRNAs. And of course, the chloroplast, can you guys see the slides?
Yes, sir.
So, and of course, this has chloroplast being maternally inherited. It has the uniparental inheritance system. So it is only being coming from the mother. So any kind of cross contamination that can happen because of the pollen grain or cross pollination will not happen in case of chloroplast transformations. It will never get contaminated, or no gene pool will come from the father side or from the pollen side. So the uniparental inheritance will helps to maintain the maintain the sanctity of the genomic content. And the chloroplast genome is not affected by epigenetic effects. So there are no epigenetic effects in the chloroplast genome. So that's why transformation of chloroplast is emerging as a very, very important and a unique tool. I've tried to list out some differences between chloroplast transformation and nuclear transformation. So you can, I'm not going to read through them, you can go through them actually. I have mentioned some of this already. Single promoter, because it's under a operon system of regulation. So single promoter for expression of multi-subunit complex protein. But here several promoters are required, and then simultaneous expression of several genes. I've already talked about it. Then transgenes is much higher to it. Transgene expression. Transgene expression is much higher. And here the nuclear genome we have already talked about. There are about 100 copies of a single, about 300 chloroplast in one single cell. And so that's why the number of chromosome is, number of DNA is, amount of DNA is more. So you can go through them in a bit more detail actually, as in when you will go through the video lecture. So I have not I have kept the slides separately. I have not included in here. Tomorrow, what we will do is we will talk about the ways of chloroplast transformation, ways of the different strategies used for chloroplast transformation. And then we will move on to concept of cisgenics and transgenics. This is another new concept that is coming up in modern day plant biotechnology. So it's important to introduce this thing to you guys. So we'll talk about, so that's why I have thought that this lecture is not pertaining to any specific topic, but all those the relatively newer aspects of plant biotechnology, which included chloroplast transformation, and of course the selectable marker genes and the screenable marker genes, and cisgenics and transgenics. So all these things will be covered in this particular this particular lecture, like today and tomorrow. So tomorrow we'll talk about that, and we'll try to cover up the cisgenics transgenics part as well. And then on Friday we'll have the discussion session, the interaction session. So you guys are required to post your videos on that, actually. Sorry, post your doubts in the group, so that I can be ready in advance. Any points? Any queries for now?
Nothing, sir.
So if there are no such points for now, I'll stop sharing the slides and we'll end the meeting now. All right.